My name is Glory, and I am analyzing myosin 2's role in sorting VPS10 and in transporting SYNC1. We know that Alzheimer's is a common disease in America. It results in the dysregulation of intracellular pathways between the endosome and the Golgi. Now we analyze myosin 2 in this experiment because myosin is involved in transportation of cargo between the endosome, the Golgi, and the transport vacuole. In using an analogy, myosin 2 would function as a truck that transports cargo throughout the highway to its destination. To better understand myosin's role in transporting cargo, we conducted two experiments. For the first experiment, we tried to see the colocalization of VPS10 GFP to see if it accumulated in the endosome or the transport vacuole. And we used that analyzing a myosin mutant strain. We used FM464, which was in red, to dye a transport vacuole. Our hypothesis for the first experiment was that the VPS10 GFP would colocalize with a transport vacuole to produce a yellow fluorescence. Then for our second experiment, we analyzed myosin 2's role in transporting SYNC1. SYNC1 is a cargo normally targeted to the plasma membrane. SYNC1PM is a mutant strain that lacks endocytosis, yet in our previous research, the exocytic pathways for both the wild-type strain and the mutant strain showed that SYNC1PM was transported to the plasma membrane, and this suggests that the effects of myosin mutants occurs in the exocytic pathways, which is also known as the recycling pathways. Yet one had to test and observe the results with other mutant strain for confirmation. So our hypothesis for the second experiment is that the myosin 2 wild type and the other mutant strains that we're going to test will have the phenotype in transporting SYNC1 to the plasma membrane. For the methods and procedures, we used the Kia Mini Prep Kit for the purification of bacterial plasmids, VPS10, SYNC1, and SYNC1PM. Then we isolated DNA and produced yeast colonies as the body for the bacterial plasmids. We then conducted one-step transformation to transfer the plasmids to the yeast cells. Under the mini prep, bacteria was grown overnight in an incubator. To obtain the plasmids, we used P2 and N3 buffer to break down the cell wall and to free the plasmid. We then used the spin column to trap the bacterial cell residue in a silicone matrix in order to isolate the plasmid. The spin column was then cleansed, and then we used an EB buffer in order to create the plasmid culture, and then we put that in the freezer. As for one-step transformation, we vortexed the yeast cells and we centrifuged doses. We then added transformation buffer with ssDNA to manipulate bacterial plasmids and then the target plasmid onto the yeast cells. After we put that culture in a water bath for 30 minutes at 45 degrees Celsius, we then put the culture in a petri dish and grew the plasmids over the next couple of days. We then conducted one-step transformation to transfer the plasmids to the yeast cells. We could not conduct any of these results because of the COVID-19 outbreak. Although we were to complete this experiment, VPS10 would accumulate and glow yellow to confirm that myosin 2 mutants transfer it to the transport vacuole at one point. As for our second experiment, we predict that the other myosin mutants would show the same phenotype as the wild-type strain to transport SYNC1PM to the plasma membrane. Understanding the location of VPS10 and SYNC1 allows us to better understand myosin 2's job in intercellular trafficking and to further understand how a defect in myosin 2 can affect the cargo. Since we knew that VPS10 is transported to the vacuole, it can help us to know the target areas of the disorder. And as for SYNC1PM, the results allow us to understand the pathways for which dysregulation occurred. And this helps in the study of Alzheimer's, as we know where certain defects occur to develop potential cures that could target areas of disorder. Thank you.